This is the tiniest motor in the universe. I mean, it's so tiny that 100,000 would fit side by side onto a millimeter. In our body, we probably have hundreds of quadrillions of these motors whirring.
How does this technology compare to anything man-made?
It basically has 100% energy efficiency. We can't get a motor as efficient as that, which leaves us in the dust.
Why can't natural selection explain the nanotech in our living cells?
The simplest living thing had to have machines to error check, to preserve DNA from being broken apart, and to detangle. Natural selection can't be used to explain this, but the Genesis account makes perfect sense of what we see in the scientific world.
All living cells have incredibly complex nanotech designed with atomic level precision. Dr. Sarfati, you have a PhD in chemistry from an accredited secular university. Could you explain what these machines are and why their complexity is so significant?
Well, living things by definition can make copies of themselves, and all our bodies are made of microscopic cells, and that's the simplest form of life is a cell, and it has to copy the information, the instruction, but also the machine has to be copied itself. So whenever it makes a copy of itself, so much machinery is needed to ensure that the copying takes place, and also the machinery is used to maintain so the cell can live and take care of itself before it has a chance to divide and make a copy of itself. So much machinery is needed for even the simplest life to work.
Okay, so you've introduced a variety of machines that are inside living cells. Specifically, I wanted to look at ATP synthase and why it is a marvel of biological complexity.
Well, interesting, when they first worked out that ATP was a miniature rotary electric motor about 1996, and the Nature article called this real engines of creation. Now, Nature is a very evolutionary journal, but interesting, they use the word creation. They can't help themselves, and the discovery won a Nobel Prize. Now, this is the tiniest motor in the universe. I mean, it's so tiny that 100,000 would fit side by side onto a millimeter. It rotates at about 10,000 RPM, and each rotation makes 3 molecules of ATP. Now, ATP is the energy currency of all life. Our own bodies will make our own weight in ATP and consume it just as fast as it's being made. Cyanide is so deadly precisely because it stops ATP being made. Okay, so it's an incredibly important thing. But now we know ATP, adenosine triphosphate, is made by the tiniest motor in the world, and more discoveries have been done about this. I mean, it uses a positive electric current, protons, while our motors use a negative current, which is electrons. So you have to have something to set up a voltage to drive the current. That's one thing that needs to happen. That's quite complicated. And then there's a conversion of electrical energy into the chemical rotational energy, and the bottom part has 10 parts. The top part has sort of 3 parts, and that sort of 10 to 3 is a bit of a mismatch, but that enables elastic power transmission, which enables basically 100% efficiency of converting electrical potential energy to the chemical potential energy of ATP.
Wow, this is incredible, complex nanotech that we've already introduced on the living cell size. And from what I gathered, there's a lot of this in a single living cell. Not just 1 or 2 of them, but several that are doing the job.
Well, I mean, in our body, we probably have hundreds of quadrillions of these motors whirring. You don't hear them very well, do you? But that's exactly what we have. Because every cell, yes, there's a power plant called a mitochondrion, and that is where the ATP motors are done. And cells have lots of mitochondria. Each mitochondria has lots of these molecules, and you got about 30 trillion human cells in a typical human body of 70 kilograms or 154 pounds. That's about 30 trillion human cells.
So how does this technology compare to anything man-made in its efficiency, and what have inventors learned from it?
So I don't major too much on the RPM because that depends on the scaling of things. So I'm not going to major too much on that. Okay, 10,000 RPM is still amazing, okay, but a hurricane might have about a quarter of an RPM or less. Hurricanes, of course, because they're so large, it takes a long time to rotate. It's not even less than that. Okay, but the thing is, the high efficiency, we can't get a motor as efficient as that. I mean, our car motors are lucky if we get 30. The most efficient sort of power plants are things like the combined natural gas cycles, where you have a steam turbine, but the heat's left over, boil some steam, so you get another form of generation. That's about 60%, but this is about 100%. So it's really quite amazing. And to try to miniaturize, because every motor has lots of different parts, and to try to miniaturize them is a hard thing to do, but all living things have incredible miniaturization, which leaves us in the dust.
Interesting. So how essential are these ATP synthase machines for life, and how do they work to maintain DNA?
Well, I mean, lots of machines are needed to maintain DNA. The ATP powers a lot of those machines, and they're involved in the reproduction of DNA, replicating the instruction, but also to decode the instructions. They're heavily involved in there. And in fact, ATP is used to make ATP synthase molecules. Okay, because you have to decode the instructions and read those instructions to make the ATP, but you need ATP to power that machinery, but you need the ATP synthase to make the ATP to power the making of ATP synthase. So it's a real vicious circle for evolutionists to try to overcome.
Do you know how long one of these ATP synthase motors is expected to last?
I don't know that, sorry.
But it wouldn't be very long.
I'm not sure how long it would last, but I think they would be constantly being made.
Interesting. Okay, so could you discuss the relationship between DNA, its repair machines, and the challenge that this poses for theories on the origin of life?
Well, essentially, you go back to 2015, Nobel Prize for Chemistry awarded to Thomas Lindahl, who discovered that DNA breaks down at a rate so fast that life shouldn't be able to exist. That's what the Nobel Prize website said about the award. So Lindahl realized that for life to exist, there must be some sort of repair machines. And so he set out to try to find out how DNA is repaired. And the two other prize winners found different ways of DNA repairing itself. But the instructions for the repair machines are on the DNA, but you couldn't keep those instructions unless you already have repair machines to stop them being degraded. So DNA has instructions for its own repair machines. It's really quite something. So this calls into question things like, how could DNA arise in some sort of primordial soup without repair machines to stop it degrading? Another thing is DNA has been found in dinosaurs. Now, dinosaurs were meant to have died out 66 million years ago, but the DNA could only last a few thousand years at most. As a DNA being discovered in dinosaur bone is a huge challenge to the millions of years view, but in fact consistent with the biblical view that dinosaurs were destroyed in the flood, or most of them were, and that was about 4,500 years ago. That's consistent with what we observe with DNA, but millions of years, forget about it.
Okay, that sounds fascinating. I know I've heard a little bit about finding dinosaur DNA in the fossils. Can you talk a little bit more about that and tell people? I imagine most people haven't heard about this. Before we continue, a quick heads up. Stick around until the end of the interview, because we'll be offering an exclusive free stream of our award-winning documentary, Evolution's Achilles Heels. You won't want to miss it.
Well, see, DNA, they've actually measured breakdown rate of DNA. In fact, I think that they calibrated moa bones from New Zealand with carbon-14, which has its own problems. So the dates are probably exaggerated, but let's grant the dating is accurate. And they found that about at 13 degrees Celsius, the DNA has a half-life of about 500 years.
Yeah, nowhere near the millions it would take for the dinosaurs.
And the thing is, if you even try to freeze it to -5 Celsius, about 23 Fahrenheit, the DNA would last a maximum of about 22,000 years. That's if it's deep frozen. Wow. But when you take it up to sort of higher temperatures, warm temperature, the half-life drops to thousands of years. In fact, they can talk about the time it takes before there's even just one thing left of it, just broken into single pieces. And it turns out that so 6.8 million years to complete fragmentation, even if it's frozen at -5 Celsius. And the dinosaur they were measuring, and one of the T-Rexes, they say it was 68 million years, so 10 times longer than the maximum survival time, and yet DNA was still intact enough to give a chemical signal. It still had the double helix structure. So it was intact. Not the whole full thing, but it was intact enough to give you a double helix.
Yeah.
And yet it should have been broken down to single pieces in a tenth of that time if it was deep frozen. And yet we know dinosaurs lived in tropical climates. That's what they tell us. They lived in warm climates where, of course, DNA would last even less time.
Interesting. All of that, incredible findings. Now, I want to return to the ATP synthase and DNA, but we should look at RNA as well. What are the challenges for RNA being the first molecule, and why is this still a popular theory?
Well, you see, life has to both have information storage and processing, but also has to have metabolism, how its chemistry works to feed itself and repair itself. So metabolism plus information. And they were hoping that RNA could do both. Hence, they thought the first life might be RNA world, and then it hijacked or co-opted some proteins to do the machinery work. Now, several problems. One is that RNA doesn't make very good machines. It's very limited in what it can do. The second thing is any given RNA molecule can be either a machine or an information storage thing, because to store information or to replicate, you have to have the strands open to be copied or decoded, but to become a machine, they have to be bound together to form the structure with atomic level precision. So it can do one or the other, a given molecule, one or the other, not both. But the other problem is how unstable the RNA is. Okay, I mentioned DNA being unstable. That was a Nobel Prize, 2015. RNA, I should define it, shouldn't I?
Yes, please.
Okay. Well, see, DNA, it stands for definitely no accident, really. But deoxyribonucleic acid. Deoxy means it's removed an oxygen. RNA just stands for ribonucleic acid, because to go from RNA to DNA, you change an OH, hydroxy, you remove the oxygen from it, it becomes just a hydrogen. But that OH makes it much more reactive. It can react with other things. It can actually react with itself and it cleaves itself, self-cleaving with that extra thing. So RNA is about 100 times less stable than DNA. And that's saying a lot, as we know. So the idea of an RNA world is preposterous because of the instability of RNA.
Yeah.
And even to get RNA from the building blocks, again, the chemistry goes in the other direction.
So what you're describing thus far is this idea that this nanotechnology in all living cells, incredibly small on the atomic level, it's got this incredible precision, and it has everything stacked against it. We have DNA that easily, pulls itself apart, unravels, and RNA, which is even more fragile than the DNA, but the information needed to create the machines is on these codes, and then it creates the machines which have to take care of each other and creating the energy to maintain each other, as well as take care of the RNA and the DNA in a single living cell. And the precision has to be so good across the board that you're looking at virtually, how many copying mistakes can it afford?
Well, there's actually research done on that. They talk about an eigen paradox. Manfred Eigen worked out that probably if you have a genome of so many letters, probably one mistake per copying cycle, any more than that, and it will collapse into error catastrophe, he called it. See, what happens, you've got DNA being decoded into some sort of copying machine. If there's a mistake there, the copying machine gets a mistake, so it doesn't do a good job of decoding, and then it introduces more errors into the next cycle, and so it doesn't take long for the whole thing to come crashing down, okay? Hence error catastrophe. And there's an eigen paradox. He said that any error-checking machine would need a lot of DNA to encode the error-checking machine, but then you couldn't copy that unless you already had the error-checking machine present.
Yes, you can't have the one without the other.
Exactly, yes.
That is a big problem. Could you explain this paradox and how it ties into the concept of life being created very good as described in Genesis 1?
See, Genesis 1 is totally the opposite of what evolution teaches us in the time frame, the details, and even the order of events is diametrically opposed to evolution as you can get. Now, Genesis is very clear. 7 times it says the creation was good, and the 7th time it was very good. Now, 7 is a number of perfection in the Bible, so God is telling us something. Genesis 1, the creation was perfect. And we see from looking at this machinery, the machinery had to be basically perfect right from the beginning, otherwise error catastrophe would ensue. So which means that you had to start life very good, as Genesis says. It couldn't start mediocre or even good and work its way up, because if it's down here, it's going to go further down really quickly. So unless it's starting up here, very good, as the Bible says, it's not going to exist at all. So, but what we observe is we can see the perfect concept of design still, but we also see the effect of sin, which has ruined it. So we're seeing things go downhill. But the evidence of the machinery shows it had to start up here and not down here, as evolution says. Evolution says it starts right down here with a pool of chemicals, right, and worked its way up here. But no, it had to start up here.
Well, what that brings to mind is thinking how evolution supports the idea of in using natural selection, that the fit is what survives. And we're describing things that are very weak at the beginning. So how could the earliest origins of life that were incredibly simple, small, weak, and helpless, their conditions were really unfit compared to anything that we have with living organisms today?
Well, see, now biologists natural selection really means reproduction of the fittest. They call it differential reproduction. So the fittest are the ones that leave the most surviving offspring. But that means what do you need for natural selection to work? You have to have something that can reproduce, because you're trying to select something, okay, but unless it can reproduce, it's not going to pass on that to the next generation. So you must have reproduction before you can talk about natural selection. And this means you can't use natural selection to explain the first self-reproducing things. Only once you got the self-reproducing things, at least 2 of them, so they can compete, can you even talk about natural selection.
So not only do you need all of the nanotech to be fully functional in the DNA, or not in the DNA, but in the living cell, along with the DNA and the RNA, doing what the living cell needs to do, you also need it to be able to create more, and for this living being, species, and creature to procreate in order to get to the next generation.
And that's one difference between living things and non-living things. None of our motors have baby motors, okay? But living things have copies of themselves.
Right. So DNA, being so long and thin, needs detangling as it's processed. How do the detangling machines like topoisomerase work to manage this?
Well, okay, see, DNA is about 2 nanometers in diameter. It's very tiny, because a million nanometers make up a millimeter, okay? A million of those is a millionth is a nanometer, okay, so 2 nanometers in diameter. But if you laid out all the DNA in one of your cells, end to end, it would be about 2 meters. Or 6.5 feet, as you would say. Okay, so very, very long and thin. It's a double helix, so it's coiled up. There are 2 strands. Now, what happens if you try to pull apart a coil? The coil behind it's going to become super coiled and resist the taking it apart. Very quickly, you won't be able to take it apart anymore. And yet, when it comes to replicating or to decoding, you only get 1 strand at a time, which means you have to take it apart. So what every living thing has is a detangling machine. The fancy word is topoisomerase. One type is called gyrase, which is much easier to say. But what that has to do is it has to cut the DNA, and maneuver it to remove a knot, to remove a coil, a supercoil, and then it has to put it back together.
Yeah.
Because you've got to have it in one piece, okay? And every time DNA is being copied or decoded, you have to have these detanglers working at that time. Now, several problems for evolution. One is the irreducible complexity, or the minimum functionality threshold. You've got to do 3 things: cut, maneuver, and put back. If it doesn't do all 3 things, it's worse than useless. Because imagine, first step, it's cutting, and then it chops the DNA to smithereens, and it's useless. Natural selection would go against it. The other thing is, because it's needed for reproduction, you can't even talk about natural selection yet. Okay, and Another thing is DNA encodes its own detangling machines, but unless you have detangling machines operating already, you couldn't decode that stretch of DNA to build a detangling machine unless you already have them there to begin with. So again, DNA has instructions for its own machines, including the detanglers.
Well, it just brings to mind how difficult it is to detangle wires, cables. I have lots and lots of those in boxes, and every time I pull them out, it's going to take me several minutes just to detangle something pretty simple for a power cord.
Right.
And you're describing that these motors and the like are actually managing this with atomic level precision on a regular basis with great accuracy, disconnecting things, reconnecting things like an electrician along the way.
Except the electricians don't tend to chop the wires and splice them back, do they?
Right.
That's the one advantage of the detangling machines themselves. They chop and then put it back. That makes it hard, though.
Yeah. So then elaborate on the implications of this for the theories on the origin of life.
Well, the simplest living thing had to have the machines I've been describing. You had to have machines to error check, to preserve DNA from being broken apart, and to detangle, and far much more, because machines are needed to replicate. And again, DNA encodes instructions for its own replication. And as I said, natural selection can't be used to explain this, because natural selection presupposes that you've already got reproduction.
Okay. Well, then while we're thinking about the complexity of life, let's also throw antibiotics into the mix. How do different antibiotics work against this, disrupting these machines? And what does this tell us about the complexity of life?
Well, a lot of antibiotics work by targeting one type of machine in the bacteria. Just one type of machine, and that seems to be enough to kill the germ, or at least to stop it from. It might actually just keep it from doing anything. Okay, it's a bacteriostatic. It means it stops it from doing anything, or bactericidal, which means it kills a thing. But for instance, the first motor we discussed, the ATP synthase, it's got two halves: an upper half and a lower half. The lower half is the electrical current. The upper half makes the ATP. But there's an antibiotic called oligomycin that binds the lower half, and they call that lower half the oligomycin binding fraction, or F0, FO for short. FO, it shouldn't be F0, FO for oligomycin, okay? And that's an antibiotic. There are other antibiotics like streptomycin, which binds the ribosome. The ribosome is the machine that decodes the protein into the amino acids, which we probably talk about, right?
Right.
But that's what the ribosome, the streptomycin, does. And there are also macrolides and tetracyclics, which somehow stop the growing protein. So it stops proteins from growing, okay? Then you got things like the fluoroquinolone, or just the quinolones. What they do is they attack those detangling motors and stop it from putting the DNA back, which means they go through the bacteria's DNA, chop to smithereens, and don't put it back together again. So the bacterial genome is totally chopped to pieces. Okay. Other antibiotics are the famous penicillin, the best-known antibiotic, penicillin, and similar things called cephalosporins. What they do is they just chop the cell wall, and if the cell wall isn't functional, the stuff leaks out and the cell basically bursts. So again, just targeting one part. But the thing is, it shows the bacteria need all these machines to survive. Losing just one of them is dead, which means the first living cell had to have all these machines to work.
If you're enjoying this interview so far, you might be interested to know that we also produce the world's most widely read magazine on the creation evolution controversy, Creation Magazine. It's packed with beautifully illustrated articles showing how real science supports the Bible. It's designed for the whole family, and it is a great tool to strengthen your faith. Sign up now to receive 4 issues a year. Follow the link in the description. Now back to the interview. So just looking again at DNA real quick before we get to the last few questions, I wanted to ask, what is the DNA doing in the living cell?
Well, DNA is the message molecule, the information storage molecule of all living creatures, okay? So it has instructions to build the proteins, the machines of life and everything else we have. But in fact, there's a code that turns the protein, the DNA letters into protein. It's actually got there are codes there, but in fact it's better than that because there are multiple codes. They've even discovered how just a few months ago about where something is located on the DNA. So it's a positional code. There are about maybe half a dozen different codes. If you think of a book, you read a book in English, one language is proof of human intelligence, but DNA is like multiple languages. Like a book you could read in English and in German and read backwards in every 10 letters and you're still getting a message from it. And that requires superhuman intelligence to have multiple codes on the same part of DNA. Okay, but the best known thing is the code to make proteins, and that's a two-step process. They call that, the first step is called transcription, and that means you're going from DNA letters to RNA letters, which are three of them are the same, one of them is different. Okay, that's transcription. It's interesting how that works. There's a certain enzyme that goes along the DNA, but what it does is it pulls into one strand. That stores up electrical energy, mechanical energy, like rubber when it's scrunched or something or stretched, it stores energy, and then it releases that, so it goes boom. Mm-hmm. But it's using the scrunching energy to do that. So it's interesting how this scrunching machine works for transcription. But then what it produces is a part of RNA, the other type of molecule we talked about. So DNA is a sort of permanent information storage. I mean, think of the instruction manual for the whole cell, okay? But you only want to read one page of it. So you make a copy in RNA using that scrunching I talked about. And then that RNA, messenger RNA goes to the ribosome, and that's decoded into a protein. And you have 3 letters of RNA correspond to 1 letter of protein, and it turns out that the genetic code, that's a genetic code, is about optimal to minimize the chances of mistakes creeping in. So it's actually designed to preserve and not to change. So that's quite interesting. It's a very stable code. Okay, and there are a lot of the ribosome itself, where this happens, is an incredibly complex machine. What goes on there, because you've got adapter molecules that connect the RNA code to the protein code, and these have got special characteristics. Getting those, and again, DNA has instruction for all its decoding machines, but you couldn't read those instructions unless the decoding machines were already there.
Incredible. So taking a step back from all of this nanotechnology, all of this has been fully functional in living cells of all the various species and kinds, going back to the last common ancestor and before. So it is believed that the last universal common ancestor was evolved during a period of heavy bombardment on Earth. Can you explain why this timing is a challenge for the theory of evolution and the origin of life?
Well, yeah, they talk about LUCA, last universal common ancestor, because it's the common ancestor supposedly of the eukaryotes, which are what we are. We've got nuclei, we've got different cell structures. That's eukaryotes. And you have the bacteria as another big major domain of life. And then you've got the archaea as the third major domain of life. And this is the common ancestor of all three, the LUCA. But now they're realizing to be the ancestor of all three, it must be incredibly complicated. And they're now saying that it was as much as 22.5 million DNA letters, coding for about 2,600 genes. Okay, that's incredible. And they're putting it back to 4.2 billion years ago. Now they believe the Earth is 4.5 billion years old. So you're talking about 300 million year difference between the formation of the Earth and the formation of LUCA, which is already very advanced. So the evolutionists tell us. Okay, and yet they believe, the geologists believe that for the first maybe half a billion years, there was a heavy bombardment. The moon's got craters, and they thought the same was happening to the Earth. It's getting bombarded, and these bombardments, of course, obliterate any life going on there. So, of course, life couldn't evolve while it was being bombarded. But now they're saying this complicated creature, LUCA, was there during the bombardment. So we've got to actually push the bombardment back, or maybe it didn't happen. You see, they're revising a lot of theories. And LUCA couldn't have been the first thing, because they believe, of course, it evolved from much simpler things, but there's then only 300 million years at the most to get up to this 2,600 genes of LUCA. So now so maybe life had to come from outside. Just not enough time to evolve on Earth, they're telling us. Some people are.
Right.
And it goes into the other things. Why would God create parasites? Okay, it was interesting because a parasite, mycoplasma, is the simplest living thing we have now. But that's only got 600,000 letters and about 500 genes. So it's much simpler than the alleged ancestor LUCA, which is much more complicated. And what we do observe is that the parasites are degenerate forms of what God created very good. And they always seem to be degenerate because a parasite gets its nutrients from whatever it's infecting, okay? So it loses the information to make them. So it means the first living things couldn't have been parasites. They had to be more complicated. So that, again, just puts up the complexity of first life even more.
Okay, so you did bring up God. You're raising an alternative. And what is the alternative that makes more sense than the evolutionary origin of life?
Well, I'd say the creation as found in the book of Genesis, which was endorsed by Jesus Christ himself, who proved his credentials by rising from the dead. And he endorsed Genesis as real history, okay? And the real history of Genesis gives us a coherent account because it tells us that God made things very good. Okay, and as we pointed out, the things had to start off very good. Otherwise, they would deteriorate really quickly. Unless they were already up here, they would die. So the Genesis account makes perfect sense of what we see in the scientific world.
Fantastic. Well, you want to unpack that a little bit more then for us? Because we've described a lot about the evolutionary origin of life and how it doesn't work. I think it would help to understand this other scenario a lot better.
Well, I mean, we see that when we want to make a machine, there has to be intelligence. When we see an instruction manual, we know there was an instruction writer. When we see any sort of book, we know there was an author of the book. But in living things, we see an author so brilliant that he can put different codes into the same letters. So what we see, it really does match up with what we read in the Bible. There's a creator of infinite intelligence, and who is the creator of time, and therefore doesn't need lots of time. And he started life off basically perfect. And what we're seeing is that after the fall into sin, things have started to go downhill, but they must have started uphill. That's what we're seeing in science. That's what we see in God's word.
Dr. Sarfati, this has been a great conversation looking at the sciences and the origins of life from evolution as well as the Bible. A lot of our listeners are probably interested in learning a lot more. So we have a free resource that we want to offer them. Can you tell us about that?
Well, thank you, Joseph. The resource is called Evolution's Achilles Heels. It's an award-winning documentary. It covers a huge range of topics. In fact, there are 15 PhD scientists. They're all speaking in their own field of expertise as to why evolution doesn't work. And the session most relevant to what we're talking about is on the origin of life. That discusses machinery, but there are also ones about natural selection, genetics, radioactive dating, Big Bang, the fossil record, and even why evolution matters for morality. For instance, how evolution was the basis of the Nazi Holocaust. And it's a very good teaching tool because it's 9 10-minute sections, so you can show it over several weeks, and it comes with a free study guide, so you can use it as a great teaching tool.
Excellent. If you want to watch the award-winning Evolution's Achilles Heels, you'll find a link to stream it for free in the description. Dr. Sarfati, thank you so much for your time.
Thank you, Joseph.